told them not to come in late. We'll see. All right, so let's talk about, guys, what we discussed a little bit last week. So I think last week we talked about transport, right? So so I think the equation that we discussed also was the water potential, right? The sonic potential plus the pressure potential. So this is one of the things I think toward the last class. This is what we talked about. Okay. So, and one of the things that we talked about is, you know, like a few things that we talked about. You know, was if you have, for example, this the water. And if you throw in a cell in there, right? So if you throw in a cell, so the understanding is because distilled water, you know, supposedly you have a water potential of 0, and then this one has a potential of negative value. So the water is going to go from the high potential, low potential. Water is going to start going in. Okay, so that's the understanding that we talked about, right? And the process of water moving based on the potential is called osmosis. Okay, that's one of the stuff that we talked about. So, so if you look at it the other way around, right? So you know, if you look at it, guys, where if you put the cell in salt water, right? Salt water, and you put a cell in there. So let's talk about let's imagine, guys, the salinity is a little higher inside the little tank compared to the cells. Then this will be more negative. For example, this is negative like 2. This is negative 1.5 or something. So then, if you think about it, what will happen, guys? The water is going to start coming out, and that's also the osmosis, right? So you know, like you probably remember, guys, back in time, you know, what you guys knew before is like, you know, back in time, right? How do they preserve like meat? Is what they used to exactly salt them, right? They salt them. And how does that work, guys? Because why? If you salt them, what happens? The water comes out, right? So water leaves the cell, and then that's basically what they turned into, like a beef jerky back in time. Because that's how you preserve it. And also, I don't know if you guys ever heard, like back in time, like Mongolians, right? When they're invading like, you know, other places here and there, what they did is made them into, you know, like, you know, that's what they salt the meat. And then later, when they go to places, they put water there. The water goes back in, and then they can make the food. You know, like based on that meat. So it's all in all, you know, the salt is there to preserve the meat based on, you know, water coming out of it. Osmosis perspective. Okay, so that's one of the things that you have to know for the AP Bio perspective as well. So AP Bio, they want you to know the equations and how does it function, right? Any questions on this one? So this is what we talked about towards the end of last class. Any questions, concerns we know? So one of the other things that we talked about, guys, is the active transport and passive transport. That's one of the stuff, guys. I think where I left off the class, I believe, was like co-transport, guys. That's one of the stuff that I left the class. I left off the class. So if you recall, guys, okay. So cell membrane. So based on what we talked about, guys, cell membrane has phospholipids, and they have these. These are called integral proteins, right? So some of the integral proteins are what we call a transmembrane. Okay, so there's you know, the opening on both ends is facing the outside and also the inside. It's called a transmembrane protein. So you know, so basically their main role is basically what, guys? For the, I mean, typically what? Transport could be one of the very main roles, right? So these proteins, like these integral proteins, has many roles, many purpose for them, right? So the little stuff we talked about. So they can be transport. That's the main thing. And also, based on what we talked about, I mean, they can act as like catalytic enzymes, as we also talked about. Okay. And it could be what, like signaling molecule, okay? So that's one of the, I mean, so I mean, this is something that we discussed last week, so I don't want to discuss all that anchorage and, you know, recognition, recognizing the other cells and stuff like that. So, you know, that's basically what these integral proteins are for, but we just want to take a look at it in perspective of the transport only for now, okay? So, so for example, guys, one of the things that we talked about, if there are more stuff on this side and less stuff on this side. So one of the thing that you'll hear a lot, guys, is a sodium and potassium pump. Okay. Sodium potassium pump. So I'm not going to get into too much detail for you guys because AP, like even AP guys, they don't talk about it too much. It's a sodium ion and potassium ion pump. So basically what they do is that for any cell, okay, they pump out sodium, okay, and typically guys, they pump out three sodium ions per time. And at the same time guys, they pump in 2 potassium ions. Okay, so they take out 3 ions of sodium and pump in 2 potassium. So this is active transport, which is at the what happens at the expense of ATP. That's one of the stuff that we talked about. So when ATP phosphorylates these proteins, I mean, they change in shape. That's why they push out the Na+. The phosphate group comes out. They change shape again, brings in the K+. So these ATP sort of trigger the change of configuration of these proteins. Okay, so 3 goes out, 2 comes in. That's something you see. So if you look at this, guys, so if you think about it, right? So there are more. So of course, like there are more Na+ on the outside, but what happens, guys? Yeah, they're pushing it more out there because what? Using the ATP. That's what we call, guys, active transport. Okay. And then the 2K pluses come back in. Okay, so, you know, that's also active transport. There's a higher concentration of K plus in the membrane inside the cell. That's also an example of active transport, which is at the expense of ATP. So, you know, again, like you guys probably understand, guys. Also, some of you guys may have a little background in physics here. So, think about it, guys. There's 3 positive charges going out, 2 positive charges coming in. So overall, as a net perspective, okay, there are more positive charge on the outside than the inside, which means what? This is more positive on the outer membrane. Inside the membrane is what? More negative. Okay, so that's why, like you probably heard, guys, like the resting potential, you know, for these membranes, guys, like it's I'm not mistaken, guys, negative like 57 millivolts or something. So you probably seen that, guys, somewhere negative 70. I forgot. So you know, so those are like negative resting potential because what? They push out more in a sodium ion compared to potassium ion coming in. Okay, that's why we have negative values. So these are called the electric potential values. Okay? Potential values. Okay. So that's one of the examples of what we call the active transport. And like one of the things that we talked about last week was like other stuff was about co-transport. That's one of the things that we talked about. So this is co-transport, guys, by the way. I think that's where we left off. And for example, guys, if there is a glucose molecule and they want to come in against the concentration gradient, for example, what happens, guys? I mean, sodium, there's more of sodium here, guys, can tag glucose and have them come in, okay, as a facilitated diffusion. So that's what we call, guys, co-transport. So it is at expense of ATP overall, you know, but again, you know, where sodium ion brings them in like this is called a facilitated diffusion, and that's one of the questions that you might get on the AP. Okay? And like there are other stuff, you know, like endocytosis, like median endocytosis, you know, but those are, I think, relatively easy that you guys can kind of catch along later. But that's majorly main understanding, guys, of the transport that you might have to know. Yeah, something that you probably already know, right? Any questions, guys? Let me know. All right, so let's kind of talk about, guys, today, guys, we're going to talk about organelles. So I don't think it's going to take the whole class, and then I'm going to move into photosynthesis and respiration, okay? All right, I think today will be the 4th week, guys, by the way. So, guys, I think, yeah, so we might have to, I don't know, we'll try to, so because I wanted to get to genetics with you guys, so we got to pick our pace a little bit, okay? So guys, cell organelles. This chapter, guys, I'm sure you guys have some background on this stuff, but I'll add whatever that's needed for you guys for entry level, as needed. But this chapter is relatively simple, straightforward, nothing fancy. So, first thing first, guys, cell organelles. First thing you have to understand, guys, eukaryotic cells, eukaryotes, versus prokaryotic cells. Okay. So a eukaryotic cell is where, of course, guys, there's a nucleus present for the cells, and prokaryotes, what, guys, there's a nucleus. You guys might have to know, you know, so not all prokaryotic cells are all the same, by the way, because there's a domain. Okay, so there's two different domains for prokaryotic cells. Okay, so, you know, so I don't think we have to discuss the domain part for AP, so, because I don't think they talk about classification that deals with domain. Okay, so I don't want to go too far, guys, on this one. But some of the prokaryotic cells resemble eukaryotes, by the way. Okay, so some of the things we say doesn't apply for all, but AP doesn't care. College board, they don't care. So I'm not gonna go too crazy, guys, here, by the way. Okay, so one thing to understand, guys, is that like they don't have a nucleus, they do. Okay, and one that's a lot of times the AP will ask you guys some of the common things about these two. Okay, so they both are ribosome. So ribosome is something that applies for both of them. Okay, because prokaryotes, guys, if they ask you guys what's the only like organelles they have, it's what the ribosome is the organelle they got. Because according to like some of the like central dogma of bio, right? What do they say? Like DNA, RNA, and protein. Okay. So that's basically what they call central dogma of bio, which means every living thing should be able to do this. Okay, DNA, RNA, and protein. Okay. So to create a protein from an RNA base, right, like messenger RNA to a protein, guys, to do this, you need to have ribosome. Ribosome is needed, guys, to create a protein. That's the reason why prokaryotic cells, which is like the very most simple living thing that we know, is what, guys? Yeah, they need to have ribosome. And ribosome is a little bit different, guys, between these two. Okay, ribosome is a little bit smaller for the prokaryotic cells, but the function is about the same. And what makes them up is also the same thing, guys, by the way. So prokaryotic cells, ribosome, or eukaryotic cell ribosome, guys, they're made up of what? They're made up of ribosomal RNA and a bunch of proteins. Okay, so that's what supposedly makes up eukaryotic ribosome and prokaryotic ribosome, guys, are RNA and proteins. So if you look at it, guys, just to let you know, RNAs, okay, some of the RNAs will have a catalytic functions. Okay, so that's one of the things that you have to understand, guys. So RNA, like their job is a lot more than you'd think. Okay, RNA, guys, means the structural unit of the ribosome, but they do have a catalytic functions, and the RNAs that actually holds a catalytic function, they call these ribozymes actually. So ribozyme, so this is something the college wants you to know, okay? So which means ribozyme is where it refers to the RNA that has what, guys? Catalytic functions that can act as enzymes, okay? And this is very plausible because back in time, right? So very primitive form of the animals, primitive form of these cells, they have to rely on the RNA for some functions, okay? So and that is, one example is rRNAs. Okay, so rRNAs can detect, some of the bases and and such. So, which you guys don't have to go that far for you guys from the AP perspective. But again, keep that in mind, guys. I think you guys have to know for the AP perspective, you guys have to have 3 RNAs, like rRNA, messenger RNA, okay, and then also a transfer RNA. So those are the only RNAs you have to know. You don't have to know anything else. Okay, so R and T. Okay. They will ask you where rRNA is being produced. So rRNA gets produced, guys. I mean, so you can't really say that for the prokaryotic cells, by the way. But again, at least for the eukaryotic cells, guys, by the way, so the rRNA production happens in something they call, guys, nucleolus. So nucleolus is actually a region within a nucleus. Okay. If this is a cell that's a nucleus, it's a little region that you can kind of visibly see. It's not a membrane bound, but you can just visibly see. But that's where the production of the rRNA occurs, actually. Okay. That's one of the questions they used to ask a lot of the SATs and stuff also as well. So, so nothing crazy, guys, by the way. So the ribosomes have got 2 subunits, by the way. So there's larger subunits and smaller subunits, by the way. So this is something to be discussed like down the road, probably not over the summer. Probably, guys, something if you guys a lot of you guys go to Jericho, so probably you guys will talk about this sometime, I think in January or February of next year. Okay, so they don't talk about it like right the time that you guys go to school, by the way. But in terms of the protein synthesis, by the way, it's smaller units, larger unit, guys. So it's something to note, guys, on these. So, but like the messenger RNA will tag onto a small unit first, and larger unit kind of sandwiches the messenger RNA. And then there's a role for this guy. There's an EPA zone. But that's a story for today. You have to know this with the APOs as well. But ribosome is a site. Okay, keep in mind, ribosome is a site for a protein synthesis. It's where the messenger RNA goes. It will attach the bone to, and then they start to translate a protein. Okay, translate a messenger RNA into proteins. Okay. So. So that's something common. So if the question is ever asked you guys, okay, so what is what will be, what do you call that, guys? Organelle that's common for both. It'll be ribosome. Okay. And now, so in terms of eukaryotic cells, of course they have a nucleus. So nucleus, just so you know, okay, it's a very large than what you think, by the way. Okay? Nucleus is pretty big, and then inside the nucleus, okay, they have, of course, like in the hurdle material, okay, which is DNA, right? Keep in mind, guys, by the way, so the eukaryotic cells, the DNA is wrapped around in a protein called histones. Okay, so imagine, guys, there's a really long rope, okay, like these wires. Okay, let me imagine these wires, right? So so it's very long. Basically what they have to do is what, guys? They have to kind of wrap it around like little like proteins, okay, and keep it that way. So those proteins are called histone. Prokaryotic cells, they, according to the College Board, they do not have histone. Keep in mind, they do not have histone, guys. Histone is only applicable for what? Applicable for eukaryotic cells only, okay? So now when DNA gets wrapped around histone, so the form of the DNA they found in the nucleus is called chromatin, okay? This applies for eukaryotic cell only. So when you find a DNA, eukaryotic cell, okay, then they call this what? Chromatin, okay? Keep in mind, guys, histone does not apply for the prokaryotic cells, by the way, which means what? Yeah, it doesn't, they don't have a chromatin, okay? So, but keep in mind, guys, remember I said, guys, prokaryotes have a different domains, okay? The main segments, guys, by the way. Some of them actually have histone, okay? But not for you guys. AP guys, they don't think histone does not apply for these guys, okay? So there's a chapter, guys, we'll talk about the cell cycle, okay? In cell cycle, there's a G1, there's S, there's G2, there's M phase, there's C phase, okay? So this is what they call, guys, the cell cycle for typical cell, okay? So now, chromatin we found, G1, okay, S and G2 phase, okay? So basically, guys, before the mitosis of a cell, their DNA we found is chromatin for the eukaryotic cells, okay? But during the M phase, it will condense into something that you probably heard before, chromatin. Okay? So during the M phase, where they have to divide, okay, what happens, guys? They use the term condensing, which I really don't like. It condenses into chromosome. Okay, and this is basically, guys, yeah. So that's only during the cell division understanding, okay? Then you probably see those like stuff you've probably seen, guys, like stuff that looks like this, okay? That's a chromosome, but before that, guys, it's just chromatin is, so chromatin, okay? So how you can think of chromatin is, it's like inside the nucleus is, so when like guys, like when you guys go to college, you guys use a communal bathrooms, right? Like, depends on what kind of college you go. So like, there's a drain, and you probably have they have a lot of hair, like like when people wash their hair, like hair falls out. So there's like little hairballs. Okay, so that's literally what the chromatin looks like within the nucleus. Okay, it's like a little threads kind of intertwined with each other, guys. It's all over the place. That's basically what it looks like inside the nucleus as a chromatins. Okay. And yeah, so a lot to discuss about the DNAs and histone and the chromatin, by the way, okay, as a regulation chapter perspective, by the way. So when is a cell, you know, for example, wants to express a portion of the DNA, express these guys like protease a protein or something, okay? Then how do they like, you know, activate that portion of the DNA, okay, or deactivate the portion of DNA? That's one of the questions that you guys might have to nail down the road, okay? So like acetylation and methylation of the DNA, and also acetylation of histone. Like that's some of the stuff that definitely you guys can know for the APs and other stuff. Okay. But for now, we're just talking about the structures. I'm not going to go too far because yeah, right now, guys, we're just you know, just skimming the surface right now. So organelles. And now. So these guys, like that's like ribosome is it, but their DNA is, they call this a nucleoid. Okay, so you know, nucleoid. So nucleoid is just, their DNA is just tangled up. Okay, so keep in mind, guys, prokaryotic DNA is circular. They have a circular DNA. Okay, so do not get it confused, guys, with the plasmid. Plasmid is a very tiny, like, you know, circular portion of DNA. Okay, they can they can interchange with other cells. But now the main DNA for these prokaryotic cells is also circular. So now this circular DNA is all tangled up in the middle of the cell. That portion where they can be found is called nucleoid. So this is a prokaryotic cells, and their DNA is all tangled up inside. That portion is called a nucleoid, by the way. Okay. But they don't have a, keep in mind, they don't have a membrane-bound DNA. Okay, so it's all exposed, and they have these specks of ribosomes. That's about it. Okay. And then, like, yeah, so this guy, like, a lot smaller compared to eukaryotic cells, and the ribosome is also bigger for them compared to size-wise compared to prokaryotic ribosome as well. Okay. Now let's talk about, guys, eukaryotic cell. You know, you guys have to know for the AP, the endomembrane system, of course. So this is called, guys, endomembrane system. So endomembrane refers to, okay, ER, which is endoplasmic reticulum, okay, and also lysosome, and then also Golgi apparatus, and also plasma membrane. I'll put it there. So these are considered, okay, what they call, guys, endomembrane system. So what it is, so like it's a fancy name for something simple, okay? So basically, guys, I mean, one thing to note, ER, right? So ER has two different, different ERs. You know, eukaryotic cell, by the way. Okay. So one of them is what, guys? Smooth ER. The other one is what, guys? Rough ER. Two different ERs. Okay. Smooth ER, rough ER. Okay. And now this is a nucleus. Okay. And now keep in mind. Okay. So with these ERs, so you gotta understand they're just a bunch of channels. Okay, they got rows, but the rough ER, okay. So it's right outside the nucleus, by the way. It's right outside the nucleus, guys, and these are little channels, okay, little sacs and channels that's right outside the nucleus. So for the rough ER, okay, they call it rough because what? They have what? The ribosomes on, you know, the membranes. Okay, these are, keep in mind, guys, by the way. These are all what, guys? ER, ribosome, ER, lysosome, and Golgi apparatus. They're what, guys? All membrane bound. They're all membrane bound, by the way. So these are all membrane bound organelles. Ribosome is not membrane bound. There's a membrane. There's no. So these membranes are what, guys? Phospholipid membrane. Okay. So the ribosome, they don't have the phospholipid membrane. Okay, but these do. Okay. So what will happen is, guys, here. Okay. So the rough ER has ribosomes on them, okay, like this. So basically, guys, a ribosome, so you gotta understand, so the location of the ribosome, okay, there are two places that you guys have to know. So one of them is what, guys? I mean, yeah, it's like planted onto the rough ER, and the other is what, guys? Cytoplasmic. So some of the ribosome just floats around the cytoplasm like this, okay? So there are two portions where they can be found. Okay, so now, you understand, guys, what they do? The ribosome rules what, guys? They are the site for a protein synthesis, the translation of the messenger RNA to a protein, right? But now, depending upon where the ribosome is at, okay, the targeting site, okay, for the protein is different. For example, guys, the cytoplasmic ribosome, right? So protein gets produced in the cytoplasm. Okay, then what's the final usage of these proteins, right? So typically what, guys, if protein is produced, okay, then what happens, guys? These proteins are being used in the cytoplasm, okay? So which means there are a lot of activity happens in the cytoplasm, okay? So one of the main things we're gonna talk about it probably later today, is what, guys? Cellular respiration, okay? Which is like glycolysis. So glycolysis is the main thing. Every cell will run glycolysis, okay, breaking down glucose into pyruvates, by the way. So this happens in the cytoplasm, okay? So which means enzyme that's associated with glycolysis, they have to be produced in what? The ribosome on the cytoplasm, okay? So you gotta understand, guys, what the hell is a cytoplasm? So cytoplasm for the eukaryotic cell refers to this, okay? Every portion outside the nucleus and between what, guys? So between the nuclear membrane to what? Plasma membrane. So every portion here is what they call cytoplasm, okay? And now the liquid portion, the fluid portion is called the cytosol, okay? So cytoplasm here, it's a region between the nuclear membrane to the cell membrane, okay? And then the fluid portion of this is called a cytosol, okay? So now, if there's ribosome on the cytoplasm, what happens, guys? Yeah, dude, then it means this protein produced will be utilized in the cytoplasm. But now, I mean, you could say like, yeah, this is in the cytoplasm too, but it's planted on the ER, okay? So typically these protein gets produced on the ribosome here as well, guys. It's for excretion, like secretion. Which means what, guys? They ship this protein out to the outside the cell, okay? So meaning when they produce this protein is what? It's meant to go outside the cell, okay, for whatever reason. So what kind of, so it doesn't have to be outside. So these protein gets produced as what? Could be what? Protein on the membrane. For example, integral proteins, peripheral proteins, whatever, okay? That could be a portion that goes out to the membrane, or what also can be the protein that goes out of the cell, okay? So why would you need the cell protein to go out? For example, guys, what? Example is insulin. Insulin is example. Insulin is what, guys? Insulin is basically it's a hormone, it's a peptide hormone produced by the pancreatic cell to signal the other cells to what? Absorb glucose. So, dude, like something like this, right? They produce it and what? They have to ship it out, okay? Because why? It's a signaling molecule, or protein, peptide hormone, okay? So and so basically, guys, like these represent the endomembrane system, which means like this kind of tells you how can a protein produced on the ER gets out. That's basically what endomembrane system is, except for the lysosome portion here. So, when a protein gets produced, so protein synthesis, again, that's a different chapter, a whole different chapter, guys, okay, very much in detail. So they produce a protein, okay, like in the rough ER. So the protein is produced and goes in the rough ER, okay? And what do they do, guys? So rough ER creates them into a vesicle, okay? So which means, guys, so this is a membrane, okay? This is a membrane, guys, by the way, so that's a protein. What happens is, they take a portion of the membrane. What happens, guys? Yeah, just put it as a vesicle, okay? So it kind of buds off, from the, ER. So like the membrane portion is what, guys? Yeah, so they take a portion of the membrane, okay? And then what happens, guys? Yeah, they put the protein produced and what? Create them into what? A vesicle, okay? And then, like, okay, so where's the next step, guys? Next stop is what, guys? Next stop is the Golgi apparatus. Okay, they have to go. That's the next stop, right? So, Golgi apparatus, guys. So that's the next stop. Okay. So then this guy will go to the vesicle will move to the Golgi apparatus. So, Golgi apparatus, guys, would be nice if you know, guys. So, this side is facing the ER, and the other side is facing the cell membrane. Okay. So this portion is called a cis portion. That side is called a trans portion. Trans and cis, okay? So cis portion is facing the ER. The trans portion of the Golgi apparatus facing what? The cell membrane. So that's called, guys, cis and trans, okay? So now, the vesicle, okay, that was created by the ER will move to what, guys? The Golgi apparatus. So, you know, like for some of the folks here, okay, you guys have to know like the clathrin. This is so these are outside the scope for the AP, okay, and the snares. So these assist, okay, creating of the vesicle, okay, and these are proteins, by the way. And snare also, you know, assists them of moving the vesicle from one side to the other. Okay, so you know. But now, like you gotta understand, guys. Okay, one thing to understand is one thing to note, guys. By the way, how do they move from here to here is a question you have to ask yourself. Okay, so how do they move from these vesicles, right? I understand. Okay, they have a clathrin coating. They have a V snare. They have a T snare. You know, nowhere to go. Okay. But actual movement of the vesicles, okay, how do they go from point A to point B within the cells? So, you know, like, okay, so do they just float around? Okay, like, how do they move within the cell, okay, from here to here? I mean, do they, like, go straight? Do they go, like, around? What route do they take is a question you have to ask yourself as well, okay? The answer is, I mean, we'll discuss this after these organelle stuff, which is what, guys? Microtubules. So microtubules, guys, basically what they call a cytoskeleton of the cell, by the way. So it provides them what? How to get to the other points, okay? So it's called microtubules. So, so these vesicles, guys, will go to the cis portion of the Golgi apparatus, and what happens, guys? Yeah, so keep in mind, guys, the membrane of the vesicle will fuse into the Golgi apparatus. Okay, so like, dude, like, okay, this is what happens. Okay, so imagine, guys, the membrane that came off of the ER will fuse onto the membrane of what, guys? The Golgi apparatus. Okay, so like it's literally what? So you understand, guys, the term endomembrane system? What happens? Dude, you can take this portion out and what? Put it there. Okay? Because what? It's the same membrane. It's an endomembrane system. Okay? And now you gotta understand, guys. So what do they do with this? So what's the job of the Golgi apparatus? So like they say, they package and ship. Okay? So, you know, so they basically, guys, so when these proteins can get there, okay, so a lot of proteins that gets out, okay, or becomes a part of the membrane, guys, those are called, usually, guys, these proteins are called glycoproteins, okay. So glycoprotein. So glycoprotein is what, guys? It's what? It's a protein that's bound by what? Bound to a carbohydrates. Okay. So this is what? Sugar portion. This is what? Protein portion, right? So it's sugar bound to a protein, right? That's called glycoprotein. So basically, what they do, guys? Yeah. So they what? They put, you know, they put a proper sugar onto the protein or swap them out to the active form. That's what they do, guys, as a term like glycoproteins. Okay, so you know. So they call like, oh yeah, modify, right? Oh, they're modifying the protein. So what do they modify? They modify what? Typically the sugar portion of the protein, okay? They put it on or swap it out, take some out. That's their job as typically your gauge apparatus. So, you can think of guys as this, okay? The ER, they typically like manufacture the cars, okay? They in terms of car manufacturing, right? And then like the car manufacturers, like after the car gets completed, what happens, guys? They ship it out to what? The car dealerships. So nowadays, these car dealerships, what? They put some modification on the cars, right? They put like more leather seats on the car. They put a different emblem on them. Sometimes, guys, I love cars, by the way. So like sometimes, guys, they tune the engines as well, guys. So they do a lot of things. Okay, so you can think of the gold apparatus as well, guys. Yeah, they like the car dealerships. Okay, so they clean the car, change some parts out. Like get ready for what? Selling. Okay, so that's exactly what's happening here. So keep in mind, guys. By the way, so a little bit detail, guys. Not that you guys really have to know this, by the way, but now. This little sacks, right? Like these are little sacks, by the way. So keep in mind, guys, they're not like tied together, which means, guys, okay, so if this portion has to go to the other portion of the gauge apparatus, they have to create a vesicle again. Okay, we'll move from here to here, here to there. Okay, that's basically, guys. Again, I don't think it's something that you have to know for AP, but just so you know, I don't even think you have to know trends and sys with AP. It's good to know. If you know this really well, if you can write this on your response or like on your school exam, like dude, like they can't mark you wrong. You know a lot more than the other students in the class, okay? So once they're nicely all packaged up, and what happens, guys? Same thing, okay? They'll butt off from the trans portion of the gauge apparatus, and what? It comes out. So now they're ready to be shipped out, okay? So now, look at it, guys, and this guy will now move to the plasma membrane, okay? And what happens, guys? Same thing, right? So basically what will happen is the membrane, the vesicle will fuse with the cell membrane, what? Will go out. That's called the exocytosis, okay? So this entire process is what they call the endomembrane system process of what? Exocytosis, okay? So they have a lot of questions on the AP like this. Okay, so like, it's very simple. It's really simple, guys. Like they ask you, oh yeah, they have a cell and like they always ask you, okay, like for example, keep in mind, guys, okay, the nucleus, okay, is sending the messenger RNA to the rough ER, okay, which is ribosomal rough ER, and now. The messenger RNA will be translated into a protein, okay? And the protein will be what? Going out to the vesicle. So keep in mind, guys, by the way, so the messenger RNA, which they got a template from the DNA, by the way, okay? So what do they do with this? Yeah, what do they do with this after the translation is over? The translation over, what do they do with the messenger RNA, guys? For prokaryotic cells, they get rid of this, they actually break this down right away. Prokaryotic cells, guys, they break it down right away. Eukaryotic cells, what do they do, guys, here? They depends on the messenger RNA, they can keep this for at least, could be a month or a week. They can keep this for a long time, okay? It depends on the cell, depends on the messenger RNA. So for example, one of the messenger RNA they talk about a lot, guys, is hemoglobin, okay? Hemoglobin, messenger RNA. They can last for weeks inside a cell, okay? They don't have to produce, they gotta reuse it for weeks. And for the eukaryotic cells, okay? So they can keep it around, okay? So protein is produced in what? Like, yeah, they follow the direction of what? Golgi apparatus and cell membrane, okay? So make sure I'm saying, guys, okay? And this, the fusing of the membrane, okay, that's what they call endomembrane system, okay? Oh. Man, I feel like it's like, there was only once in my life, guys, that I pull an all-nighter. I think twice or something. That's a freshman year in college, actually. It's the only time I pull an all-nighter. There's a, man, I never had like 2 hours of sleep in my life, actually. I tell you, man, there's a class, guys, I tell the kids, guys, there's a class called art history that I took. You guys know what art history is, guys? You know what it is, art history? So I don't know, this guy, when you guys go to college, right? All these people like try to pull you into like take their classes and stuff, upperclassmen and stuff. And now, art history, they said it's an easy class. Yeah, someone told me like it's just super easy. It's none of the things I mean, all you do is they give you, I mean, so you gotta, the class is a Monday, Wednesday, Friday, okay? You go into this like a big auditorium, okay? It's a big class, okay? And then they got these projectors, okay? And then they show you like just slides of art, okay? And mine was like Roman and Greek and like I forgot. And then you have to memorize. They tell you, they don't even like give you like a write up. You have to they will just like verbally explain. Someone comes out, I mean the professor comes out, explain what era it was produced and the time, like year, and then the name and the name of the artist, and also their meaning, okay? But in one class, they give you like 20 of those, okay? They explain, oh, that's what it was, 20 of them, okay? Monday, Wednesday, Friday, okay? But the problem is, guys, if you guys go to college, okay, it's a little different from high school. There were only 2 exams, 2 tests, that's it. First semester, guys, 2 tests. Okay, 2 tests will decide, nothing else will decide your grade for the semester. Okay, what do you think the test is? You have to memorize all of them. So each day, it was like 20 pieces of art, right? So imagine, guys, in a week, it'd be 60, 50 to 60, okay? And how many weeks in a semester, guys? I forgot how many, whatever, okay? From like, so you have to memorize hundreds of these art pieces, okay? Midterm was okay. Midterm was what, guys? Yeah, only half, right? Final is what, the whole thing? So man, I tell you guys, like that's the only time that I didn't really sleep that much for that class, and I feel the, you know, dude, I tell you, man, I can tell you that's the only A- that I got in college. Actually, that's the only class that I got an A- in, actually, because I think even the professor actually, you know, gave, you know, kind of, you know, made me go off a little easy because she knows I tried. But dude, I tell you, man, like that is something I will never do. But other than that, man, like the least amount of sleep that I got is today after that time. So yeah, so that music thing, man, I tell you, like this. It kind of reminds me of that time. Oh my goodness. And then the final was, like the test is what? They show the slide, you have to write. You know, like essay. Oh my. Yeah, don't take that class, Luke, huh? Oh my goodness. All right, so the one of the things to talk about, guys, is lysosome. This guy, okay? Lysosome. So basically, guys, I mean, it is part of the endomembrane system, okay? And now this is like, you know, this is very like high level, so you know. So it's a membrane-bound organelle, okay, phospholipid, right? And now what they have is what? They have a hydrochloric, so this is what they say. They have a hydrochloric enzymes. Okay. Hydrochloric enzyme. So what does it mean, guys? It's what? It's hydrochloric is what? Hydrochloric is what, guys? HCl. Okay. So enzyme in what? Hydrochloric solution. That's what it is. Okay. So basically, what it is, guys, inside there what? Like you have an acidic environment with what? Enzyme that works well in the acidic pH. Okay. So they're very high level, by the way. So it's not like only one of them. There could be what? Many of these lysosomes with different enzymes in it, right? So these lysosomes, what do they do, guys? So what's their function, basically? What is that? Their function is, so the understanding is, okay, it's kind of the opposite if you think about this, okay? So a cell can bring in, okay, by endocytosis. Like, you know, it could be what, guys? It could be a toxin. It could be, you know, like a food, you know, vacuole they're creating here. So what happens, guys? They have to digest it. Okay. So basically, what happens is these lysosome with hydrochloric enzyme, the vesicle and lysosome can what? They're gonna fuse together, okay, and turn into one vesicle, what happens, guys? The enzyme will start to digest, okay, whatever that was what came into a cell. So this is how, just like you know, the like you guys know, guys, for example, immune system, right? Immune system, you probably heard, guys, the immune system has a first line of defense, second line of defense, and third line of defense, okay? And now the second line of defense is what, guys? These neutrophils, some macrophage, they engulf something that came into a cell, into a body, right? And they come in like this, and what happens, guys? The hydrophilic enzyme will what? Will disintegrate them. So that's basically how the lysosome work. It fuses with the vesicle, and the fusing portion is what, guys? They can do it because why? They're part of the endomembrane system, okay? That's the understanding. And also lysosome is what, guys? They can act as what? The programmed cell death as well, just so you know, lysosome, right? So if the cell wants to kill itself, then what? Lysosome will fuse into like portion of the cell and will kill itself, okay? So these are, guys, portion of endomembrane system, and one of the things that we didn't talk about is what smooth ER. Smooth ER, guys, is basically the ER without the ribosome, okay? And typically their function is what, guys? Their function is detoxification. Like for example, like, you know, like liver cells detox the alcohol, for example, okay? So which means what? Typically the smooth ER functions inside further detoxification. And also what, guys? Lipid production. So for example, guys, the cell membrane, right? The phospholipid, they can be produced in a smooth ER, okay? And then what? Gets shipped out to the cell membrane, okay? That's basically what they call, guys, smooth ER, okay? The function of the smooth ER, detox of lipid production, like steroid molecule, right? Any questions from here? So the other things that I'll talk about, guys, is the cytoskeleton, okay, which is the part of the AP also as well. I'm gonna be pretty quick. So one of the things that we talked about is what? Okay, how can vesicle move from point to a point within a cell? Okay, so scientists they actually thought that what? They're just what? They're like a little bubble. Okay, they can move around. Okay, but is it so? Is it like, are they actually a bubble that can just like, you know, just float around within a cell, guys? Not true. So what they found is what? The cell has what? These cytoskeletons, which is made up of microtubules, microfilaments, intermediate filaments, okay? So for the AP, I don't personally think like you guys have to know like super crazy in detail, but you definitely have to know though. You definitely have to know what it is and their functions and stuff like that. So, so microtubules, okay. So, and microtubules, again, microfilaments and intermediate filaments, right? Microtubules are created, you know, from a protein called tubulins. It's good to know, okay? If AP ever wants to question, okay, oh yeah, what is like, they gotta explain microtubules, they gotta give it away. Tubulin, like tubulin is a protein that is, you know, it's a protein that, you know, is a structure of the microtubules, okay? So microtubule is what? It's a hollow little tube, okay? That's created by like, you know, tubulin. It's like a little straw, okay? Very thin, like, you know, very small diameter, by the way, okay? And these are all over the cell. These are all over the cells, okay, by the way. So they're like all over the cells. So basically, just so you know, okay, these microtubules, okay, it acts like a little rail, okay? They're like a little rail, okay, on the inside of the cell. So now, if a vesicle wants to move from point A to point B, they follow the microtubules, okay? So keep in mind, guys, they have these things called the motor proteins. Okay, they got something called the motor proteins, okay? So now, these motor proteins at the assist of what? ATP, At the expense of ATP, they're able to move from one to the other. Okay? And that's how they move. The vesicles can move from what? For example, like, dude, Golgi apparatus to what? I'm sorry, the roughly arced Golgi apparatus. Okay? They follow this what, guys? Microtubule, and these guys have what, guys? Motor proteins on them. So we know. So that's one of the usage of what, guys? Microtubules. Okay? And so you gotta understand, guys, like they will talk about motor proteins. Okay, so these are the proteins that can assist in what? In the moving of a vesicle, like not only vesicle, but moving of what? From a particle from one to the other at the expense of ATP, okay? And also, guys, microtubules also forms flagellum, and also cilium. So cilia and flagella is what, guys? It provides them. It can provide, guys, movement for the cells. Okay. So cilia, like for paramecium, right? You probably like when you were a little kid, guys, taking bio, you probably have seen what, guys? Like, oh yeah, paramecium, right? So they got these like cilia, okay, on the cells. You probably have seen this, guys. They gives them, they're able to move around from point A to point B, right? They can like make them move, right? So these cilia or flagellum, like flagella, right? They assist what, guys? The movement of the cells, and now they are made up of microtubules. And some teachers, they talk about this a little more in detail, just so you know. So yeah, I gotta use the bathroom, guys, by the way. But yeah, give me a second, guys. You guys, I don't know. I sent you guys reading, by the way, okay? So make sure you can take a look at it right now, but give me like 2 seconds, guys, okay? Okay, so let's talk about, guys, the flagellum and cilium, okay? Flagella, cilia, whatever we call it, okay? So let's structure up. So this might be a little bit too much for some folks, and also I don't know, guys, if this is a portion or the detail that you might have to know for the AP. Okay, but now it's good to know. All right. So cilia and flagella, okay, they are created from a tubule and microtubules, okay, and okay. So if I cut this, okay, cross-sectionally, okay, so there's a microtubule, I cut it, okay. How would they look is a question, right? They follow something they call what? 9 plus 2, okay? The formation of microtubules. So it's good to know for some of the folks here, guys, which means, guys, so the microtubules, okay, 9 of these guys, okay, so I'm not gonna count, 9 of these guys, they are a portion of the outside like this, okay? They're double, like one microtubule, the tube, and two of them kind of holding together, okay? And there are 9 of these guys. And in the middle, there's 1, okay? So that's what they call 9 plus 2 formation of microtubules for flagellum and cilia. Yeah, keep in mind, guys, okay? This only applies, like this thing, guys, right now, applies for what? Eukaryotes only, okay? Prokaryotic cells can have a cilia and also flagella as well, okay? But they don't have this. They don't have this formation. They're produced by completely different formation, by the way, okay? And now, one thing to note, guys. I mean, these microtubules are connected by a protein called dyneins, okay? So these proteins are like a motor protein where what, guys? They can slide along both microtubules and make them what, bend and go straight and stuff like that and give them movement, okay? So that's what this protein, so they have these proteins between these microtubules, okay, that connects them, okay? They made them able to move around like that, okay? So but that's the formation, guys, of 9 plus 2 formation of microtubules for flagellum and cilium, okay? So and keep that in mind, guys. There are some cilia even in your body that is not meant to for what? Not meant to be for a movement. Okay, I have to where in your body have a ciliated cells that is not like, dude, the cell in your body, okay, they're not meant for movement. You know what I'm saying, guys? They're part of your, like, you know, endothelium, okay? But they're not gonna move from here to here. So those cilia is what? Yeah, what are they typically for? Like, you know, in your nasal cavity, for example, okay? What are they for, guys? To make sure the dust particles are not gonna go in and, like, you know, like make sure it catches it, you know? Those cilia, which is non-motile. It's good to know, like, dude, it's good to know, okay? So would your AP ever have it, guys, or your teacher? Who knows? If you guys go to Jericho, they will never talk about it, by the way. But it's good to know, okay? Non-motile means what, guys? They only have what? 9 plus 0, which means they don't have an inner portion of it, okay? So non-motile versus motile, guys, is what? Like, inner portion of the microtubule is missing, cross-sectional view perspective, okay? So it's good to know, okay? But again, like, but I don't know, some teacher will talk about 9 plus 2, the formation of these microtubules. It's good to know, okay? Your teacher might bring up, might not bring up, whatever, okay? The more detail you know, guys, the better it is for you guys, okay? So that's the, you know, microtubule formation, by the way, okay? And now, you know, so there is something they call, guys, microfilaments. So microfilaments is created by a protein much smaller, and this is much thinner, called the actins. Okay, actin is a protein. So yeah, this guy is a tubule, right? This guy is created from something called the actin. So actin, the example I can give you guys for the actin is what, guys? Actin is a part of what, guys? Your muscle. Your muscle is produced. The very small functional group of the muscle is called what? Sarcomere. You'll never have to do that for your AP. AP, guys, they took out the entire physiology. So actin and myosin, okay, that's the smallest subunit called sarcomere of your muscles, okay? But now, actin, that actin, right, is what, guys? Plays a role in microfilament, which is also cytoskeleton. It's all over the cell, okay? Give them what? Structure of a cell as well, okay? And they play a role also in what? Cell division as well. Okay. It gives them a structure of the cell, like how they look. Like, for example, your body has a skeleton as well, right? Your body has what, guys? Endoskeleton, like the bones in your body, right? But these guys are not bones, but they're what, guys? Cytoskeleton, like they give them the shape of the cell and structure of the cell as well, okay? And they play a role in the cell division also as well, okay? It's good to know, okay? And there's intermediate filaments. Intermediate filament, guys. So this is created from multiple different proteins, by the way, okay? So intermediate filaments, guys, their role is the anchoring of the organelle, okay, within a cell, anchorage of the cell, okay, so that the organelles don't move around from here to there, okay? So they made them there, keep in place. They're not like, you know, floating around, but they're more for the anchorage perspective, by the way. So, but they don't play a role in, like, you know, these guys, okay? Like actin or myosin, like they don't like, so actin and myosin is what? The muscle cell, they move around, right? So, but that's more for the anchor to the cells, okay, organelles within the cell, okay? So these are three things. I'm not going to go crazy with you guys, by the way. So three micro, you know, the microtubules, microfilaments, intermediate filaments, guys. These are called the cytoskeleton, what compose of cytoskeleton of the cells, okay? And now, the other things you might want to know, guys, the other portion of the organelles in the cell. This is something they don't really talk about. Okay, let's talk about, guys, three more things to discuss in the cell, guys. Centrosome, okay, and the mitochondria and chloroplast, okay? There are three things left for this chapter. I mean, again, like, this is nothing crazy, guys, okay? Centrosome is a region of the cell. Region, like there's a location of the cell. This is a cell, right? We have to talk about the cell wall, by the way, for the plant cell. So this is nucleus, okay? So outside the nucleus, there's a region called centrosome. So there are two centrosomes, okay? So within the centrosome, for this is something they call, guys, animal cells. Okay, like cells that like, you know, for cells like for us guys, right? And central sum. So central sum inside Joseph guys, they have something we call centrioles. Okay, they got two centrioles. Okay. So these centrioles, okay. So the centriole, when you look at centriole, guys, okay, they have, okay, so I can't draw this very well, by the way, okay. Centriole, if you cut the centriole, guys, like cross-sectional view perspective, they look very similar to microtubules, okay, but they have three microtubule, you know, like tied together, tubelets tied together. So that's what they call like 9 of them, okay? 9 of these guys. So that's what they call centrioles. So they got 2 of these guys like that, okay? So centriole for the animal cells, they you know, not like 100%, like they think centriole is a portion of a cell where they manufacture microtubules. Okay, so microtubules that's in the, around the cell, the tubulin, you know, stuff like that. So they think centriole is where they can manufacture the microtubules. Okay, because what? They're the same thing. A little different structure, by the way, but they're the same thing. And now centriole, or like, you know, this plays a role in what, guys? The cell division as well, which is why it produces some microtubules for the cells to separate the chromosomes, for example. Okay, so it's good to know. Okay, but plant cells, they do not have a centrosome. They do have a centrosome, but they don't have a centriole. That's one of the things to know, guys. The plant cells, they do not have this centriole. Okay, but they have a centrosome, and there's no problem for them to produce microtubules. So, which means like this might not be like something that they need for the plant cells. Okay, but again, it's good to know. Okay, centriole. So like, don't get confused, guys. Centriole is only thing that can be found for the animal cells. Plant cells don't have it, okay? I mean, it's good to know these guys, like have understanding of it. I mean, even like you might not remember, but if your teacher talks about it, oh yeah, like this guy told me about this, you know? So it might help you guys down the road. And then the couple of things, guys, which is mitochondria and chloroplast. And also peroxisome. So these are the 3 organelles. Keep in mind, guys, these are membrane-bound organelle that is not part of what, guys? This is not part of the endomembrane system. Okay? Which means they're not going to fuse with the other, like, you know, organelles in the cell membrane. Okay? So mitochondria and chloroplast. Okay? Keep in mind, guys, like they ask a lot about these 2, okay? And not because of their functions, but because of their origin, okay, where they came from, okay? Mitochondria, keep in mind, guys, they got their own DNA, okay? Like they have their own DNA. Like this guy too, there's a DNA themselves. They produce their own proteins, okay? They replicate on their own within the cell, and their membrane is different than the membrane of what? The other portion of the body, other portion of the cell membrane. So, and also, guys, okay, so basically, guys, what they think, because they have their own DNAs and stuff like that, guys, like they replicate their own. So what is it? If you read it, guys, endosymbiosis, right? Which means what, guys? These guys were once a tiny cell of their own, but engulfed by the larger cell. Okay. So they become like a small organelle. Like for example, guys, you know, there's a bigger country, okay? This small one becomes part of what, like the larger country, right? Something like that, okay? But they keep their, like, you know, like their whatever, like they used to do, like, you know, their cultures and all. They keep it like something like that, okay? So there's yeah. So keep in mind, guys, okay? Like we talked about this a little bit, right? In terms of evolution. Okay, so again, like I'm very, like not like religious guys, by the way, like I'm not like whatever, you know, like not that if you guys believe in like God produced everything, whatever, okay, I don't care, okay, but this is just based on a theory, okay, based on a fossil records, okay, keep in mind, yeah, fish, like this is what the evolution says, right, Fish evolved to amphibian. Amphibian evolved to reptile. Reptile evolved to the mammals. Okay, that's basically what they say in evolution. Okay, yeah. So when did fish become amphibian based on the fossil record? 350 million years ago. Okay, so when do you think this happened? Endosymbiosis. When do you think this happened, guys? Yeah, billions years ago. Billions. Were you around billions years ago, guys? Yeah, were you? Yeah. How do they know this? It's a theory. They can't prove this. You know what I'm saying, guys? Here, it's based on a theory. Like, oh, they can't say like, oh, put the foot down. This is what happened. They can't say it because why? Yeah, a billion years ago. How can they be for sure? They can't. So, like, dude, like, okay, so they think, okay, oh yeah, they're once a cell of their own, but engulfed by the larger cell, and that's about it. You know, like, okay, so that's what they think, okay? So, and they become a crucial member of the organelle of a cell, like, portion of a cell, but they leave it at that because, but now, their function is, okay, so they ask a lot of these questions, okay, so, oh yeah, the DNA for mitochondria, it's similar to the prokaryotic cell's DNA, like blah, 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 the sequence and stuff like that, so they continue to bring this endosymbiosis theory of the mitochondria and chloroplast within larger cells, okay, so which means, like, what they're thinking is, prokaryotic cells, right, which is like, these guys were once prokaryotic cells, okay, which were engulfed by the larger cell, okay, and what provides them some, like, benefit to the larger cell, and they stayed that way, okay, and one of the things they utilize, what? Oh yeah, they got their own DNA, they replicate themselves, okay, they produce their own thing, what? They can produce their proteins, they got ribosomes in there, okay, which is what components of what? Prokaryotic cells as well. And also, guys, by the way, so these guys, by the way, so we can talk about it a little bit more, guys, they have a dual membrane, okay, they have a two membrane system. Like, for example, mitochondria, right? This is one of the stuff that some of the folks we discussed. So these mitochondria, so even for chloroplast, which we'll discuss, yeah, I do, like, my drawing's not the best, by the way. Like, when you see when someone, like, dude, like, when you guys, like, see people draw mitochondria, what do they draw, guys? Outer membrane, inner membrane. They draw the two, okay? So, okay, so this is like the cell membrane, that's membrane, okay? This is the mitochondrial membrane. This is what? Inner membrane called crystal membrane, okay? So, yeah, how do they have two membranes? Prokaryotic cells do not have two membranes, okay? How do they have two membranes, guys? So, this is what they, like, this is, guys, what they suggest you, okay? Which is, there's a prokaryotic cell here, there's a larger cell, okay? That's a prokaryotic cell, right? And now, how do these guys come in? How do they come in, guys? This is a cell membrane, guys, with a larger cell. How do they come in? Endocytosis, right? Like a vesicle. So what happens is, they turn to a vesicle. What happens, guys? Oh, this is now the membrane of this guy. Do you understand what I'm saying? So that's why they end up with what, guys? Dual membrane. Do you understand what I'm saying, guys, here? So, it's a theory, okay? But they're saying, okay, because it has dual membrane, it means what? It came into a cell by endocytosis, larger cell. Because why? These guys have what? Dual membrane system. So what's a role of mitochondria? What do they do, guys? I mean, like, very high level, what do they do, guys? They mass produce ATP, okay? Production of ATP more efficiently, okay? Chloroplast applies usually what, guys? Plant cell. Plant cells have chloroplast for what? Production of. Chloroplast, they do not produce glucose. The final product, if you guys do your reading, guys, okay, sometimes it's called PGAL. And they probably don't use PGAL in bio-AP, okay? They use the term G3P. That's the final product of the chloroplast Calvin cycle, okay? Glyceraldehyde 3-phosphate, okay? I think this represents, guys, like glyceraldehyde, L, I forgot what L stands for. Okay, aldehyde, L, I don't know. So, so they're the same thing, okay? But I think PGAL, guys, like, this is a term they use, guys, when I was in, like, high school back in time, like, studying for bio. But nowadays, a lot of books do have a G3P, okay? Glyceraldehyde 3-phosphate. This is a final product, okay? They produce this, which can be converted to glucose. So it's a special function for these two guys, okay? So that's why we use them for the eukaryotic cells, okay? So more to come on these two guys. There's a whole chapter on those two guys by themselves. Let's talk about the peroxisomes. peroxisome. So peroxisome, guys, it's an organelle of their own, the membrane, and their role, guys, is detoxification by dehydrogenation. So it's a fancy thing. So they don't want to talk about this much. So there's a very limited question on these guys, peroxisomes. Okay. So now, peroxisomes, their role is, okay, is dehydrogenation, which means what? So like the name, right? It takes the hydrogen away from a particle. So that's more for a detoxification perspective, detox. So I'm not gonna get into details, guys. By removing a hydrogen group, that makes them less, make them more polar, okay, which makes them, the enzymes can go in and what, start attacking them, blah, blah, blah. So whatever, okay? Detoxification by dehydrogenation. So what you have to understand, guys, if you dehydrogenate something, okay, you're like, what are they gonna do with hydrogen? Yeah, hydrogen, what are you gonna do? What are you gonna do with hydrogen? So what they do is what? That's not a balanced equation, by the way, but they turn into what, guys? Hydrogen peroxide, H2O2. So you probably heard this, I don't know, you probably heard this, guys. Think about it, guys. Peroxisome, peroxide, you know. So they turn into what? H2O2. Hydrogen peroxide, which is what? Dude, hydrogen peroxide kills a cell. That's toxic for the cells. Okay, will kill it. So what happens, guys? They have an enzyme called catalase. This is something you might want to know for the AP. It's called catalase. Okay? Which what? Which will turn H2O2 into what, guys? Into H2O and O2, guys. Okay? Which kind of not neutralizing, but detoxing them. Okay? That's what they call catalase as an enzyme. So, you know, like when you talk about peroxisome, I don't know, you might want to understand the term, guys, catalase. Okay, which converts to H2O2, which they produce on their by themselves by dehydrogenation, and what turns into H2O2. Okay, it is not part of the endomembrane system. Okay, it's on their own. Okay. So, we're gonna jump into these two guys, by the way, for the next chapter. But now, before we go there, guys, let's talk about cell wall and some of the transport and the between the cells for the animal cells and plant cells. So, dude, I tell you, man, your teacher cannot get any more detailed than this. So, you know, like especially like where it's Jericho, like now. So, so guys, let's talk about, guys, now. Let's talk about the cell wall. Okay, this is one thing that we talked about during the water potential stuff, right? So typically, right? Yeah, think about it, guys. Yeah, if you think about it, right? If cell that's a unicellular, whether it's a prokaryotic cell or eukaryotic cell, like it's a cell that lives by itself, okay? Like paramecium, whatever, like amoeba, you know, like bacteria, whatever, okay? They live by themselves, okay? So if you're a cell, okay, what is the worst nightmare for them? What do you think? Yeah, what do you think? What's the worst nightmare for these cells? What do you think? If you're the cell, what do you think? Yeah, what do you think, Daniel Kim? DK, what do you think? Huh? Yeah, what do you think? Yeah, what do you think?
Bursting.
Because yeah, bursting. Yeah, when will they burst?
Hypotonic.
Huh?
Hypotonic.
So yeah, so dude, exactly. So it's what I think. When it rains, dude. When it rains, and think about it, guys. When there's a rain, what happens? Rain is what, guys? Rain is like almost like what? Low, like or high, like high potential water. Okay, which dilutes the water. So I mean, what do you think will happen? Should they all die? Do they all die? Like water, oh, rain, oh, bye-bye, guys, I'm going, bye-bye. Lice, do they die, guys? Yeah, is that what happens? Are they dumb? They survive billions of years. What do they have, guys? A lot of these unicellular cells, guys, they got what? They got cell wall. They got cell wall, man, okay? To protect them against what? Lysing. Okay, so cell wall is what, guys? Cell wall is a little casing around them. Like if you got your mom got you a sandwich, so you put it in the backpack. Okay, so you'll go squished by the books and stuff like that. So okay, so your mom's like, okay, so let's put a little plastic casing around them so it doesn't get bounced off of their stuff. That's what it is. Okay, so cell wall, guys, for prokaryotic cells. So their cell wall is made up of what they call peptidoglycan. Okay, it's called peptidoglycan. So peptido, like peptido means what, guys? Like protein, right? Glycan, which is sugar. So their cell is what made up of peptidoglycan. This is SAT stuff. You should already know this. Okay. So they have a cell wall. Okay, which obviously you know the reason why. Okay. And also, guys, so these. Also, what is cell wall, guys? Fungus, they have a cell wall too, which is what made up of what? Chitin. Chitin is also what, guys? It's like it's a sugar, with some proteins on them, guys. Chitin. They said chitin is what? Cell wall. Okay. And also what, guys? Like the exoskeleton for what? Insects. They have what, guys? Chitin. Okay. So that's a cell wall the fungi have. Plants. They got what? Cellulose. Cell walls. Okay? So these are the cell walls that you guys have to know for your AP. Okay? And know their functions as well. So think about it, guys. So the water comes in. What happens, guys? These cell walls can protect them. Okay? So I don't want to talk about, guys, too much of the cell wall stuff, by the way. But now, like plant cell wall, guys, cellulose. Okay? We talked about it, guys. Cellulose is what, guys? Beta glycosidic 1,4 linkage, right? Like a beta glucose. We talked about it, right? So that turns cellulose, right? So which we can't really digest, okay? So basically, guys, what you have to understand is what, guys? The cell walls have what, guys? So primary cell wall. So this is where, guys, AP is not going to really talk about it. They got a primary cell wall, secondary cell wall, and then what? Plasma membrane. Okay? So again, primary cell wall on the outermost portion, okay? And your mom's like, oh man, like he still crushed his sandwich, so I'm gonna put one more casing, which is what? Secondary cell wall. Okay, 2 casings. So primary cell wall is relatively thin, it's a very thin cell wall. Secondary is where they can get a little thicker for the plants, okay? And then what happens, guys? And then you have what? Plasma membrane. Okay. So even if the water comes in, okay, these cell walls can provide what? Pressure potential, okay, which can what? Fight off what? Water coming in by osmosis. We talked about this, okay? And the plant cells, guys, they have a chloroplast and also they have a larger vacuole. They have a large vacuole. They have a little empty space in the middle, by the way, okay? A large vacuole can what? Like give them a storage space within the cell. Okay. That's something you have to know about cell wall, by the way. So, what about the cells who doesn't have a cell wall but they're unicellular? Paramecium. Okay. What about them, guys? Paramecium. They got no cell wall, man. Okay, but they can move. But when it rains, I mean, it's all over the same thing, right? So then, water comes in. What happens, guys? They got what, guys? You probably have seen something like this. What do they have, guys? They have what? Contractile vacuole. Contractile vacuole. Okay. So again, like if you guys want to know something crazy about it, guys, what do, guys? They pump out the water. So if you put the cell in a place where this is what? Hypotonic. What happens, guys? They what? They pump out the water. Okay. So that what? It prevents it from lysing. So AP will ask you about the contractile vacuole stuff, okay? So you put a cell into hypotonic solution with lower potential, what the hell is going to happen and stuff like that. Well, higher potential, what the hell is going to happen and stuff like that, okay? Is it more active, less active? Those are the questions you might get with AP. Very simple stuff, okay? Yeah, believe it or not, I'm not kidding. This is where the potential stuff, some students really struggle with this in the AP. I'm not kidding, guys, okay? So they forget, oh yeah, I know, and then they turn and try to solve a question, they get it wrong. Okay. And then let's talk about, guys, a little bit of a cell communication, guys, okay? So, plant cells, they're tied together by a cell wall, right? This, I mean, again, like I hate the fact that they got rid of SAT because SAT, at least you guys learn a little more about it, but so the cells must communicate or even what? Exchange materials with each other, okay? So now, if you think about it, guys, how do they communicate? Because they have a hard casing cell wall around them, right? How do they communicate? So there is an opening between the cells of the cell wall, the plant cell, for example, right? And now they exchange through these holes, okay? That method is called, this is something you have to know, guys. Plasmodesmata. It's called plasmodesmata, okay? That's where the plant cells can what? Exchange like stuff like, oh yeah, like signaling perspective, right? Like remote signaling. So yeah, we send them this and that, guys. It's by what? Method of what we call plasmodesmata, to the holes between the cell walls. That's something you have to know, okay? And now, the animal cells, I mean, they don't have to really kind of anchor them together because the cell wall anchors them together, right? But now for the animal cells, they got a little more stuff. The animal cells, for example, guys, the animal cells they don't have a cell wall, right? So they have a few things that anchor them down together, and also for the transfer perspective as well, guys. So a couple things, okay? There's something that you have to know, guys, something we call tight junction. So think about it, guys. If you drink water, okay, the water stays in what, guys? Your alimentary canal, okay? Which means like the esophagus, your stomach, your small intestine, it stays there. It doesn't leak out, okay? Why? The epithelial cell, okay, of your canal is what? The cells are tied together by a tight junction, okay? So the water cannot seep through between the cells, okay? That's called tight junction. You gotta know this, guys, okay? I made a little note here, because I forget sometimes, right? Like there's a protein that makes them, like yeah, go back a couple more weeks back. Tight junction, guys, okay? So like the protein that's like making it happen, guys, occludin, whatever. So I don't think it's know them for the AP, but so that's what tight junction is, guys. So again, like there's a protein that's involved in this, guys, by the way, like kind of zipping them together, guys, okay? And now, tight junction, guys, and also, just like the plasmodesmata, okay, there is a little hole between the two cells, guys, by the way, for the communication perspective between the animal cells, okay? And that gap is called the gap junction between the cells. Gap junction is like what, guys? Plasmodesmata for the plant cells, okay? So through the gap junction, what happens, guys? Things can what? Pass through with each other, okay? For the communication perspective. You gotta know these guys, by the way. Either one more for the AP, the anchoring of the cells, okay? The two cells, like they have to anchor them down so that they don't move around, guys, or come apart. That's called desmosomes. So you gotta know gap junction, desmosome, tight junction for the AP, okay? There are three things you have to know for the AP, okay? Their functions and what they do, okay? Like nothing more than that, like what produced them, you know, like desmosome is like created from like something, you know? They're not gonna ask those questions, okay? Like the microfilaments, by the way, they're not gonna ask those questions, okay? But you gotta understand these rules. Definitely, yeah, plasmodesmata, tight junction, gap junction, desmosome. That's a part of this chapter. Any questions on this? All right, guys. So I'm gonna talk about, guys, now the next chapter, which is there's a homework assigned for you guys, which is due next Saturday, which will be organelles and the cell transport. Okay. I think this time it'll be a lot of questions for you guys. I'll try to trim it down to less than 50. I'll try. But there'll be homework assigned, guys. Make sure you guys do it by next week. All right. So which part of that's not in the homework, guys? I'm gonna talk about it now, which is respiration and photosynthesis now. Let's talk about, guys, photosynthesis. So I think Jericho, they do this first, and then go to respiration. Okay. So I'm not sure why. They should do respiration first, but I think you guys do this first. synthesis first. Okay, photosynthesis will come first for you guys. I don't know these. I don't know like the Columbia Prep or something. I don't know what they do. So. When like what are you like sophomore? Turning sophomore?
Junior.
Turning junior?
We did cell respiration. Just grade 5.
Mm.
So I used to like
Do you know Jack? Jack.
oh, is that in the law?
Huh?
Is he in the law?
Yeah. Jack. 쟤 뭐더라? 이름 last name 까먹네. I still talk to him actually. He went to Stanford from there actually. He's 어디더라? Jack Yuan? You know? You look it up. He's probably there.
I think, wait, I'm not sure.
Yeah, like. Yeah, I don't know. I think he's in his second year in Stanford, or turning his third year. I don't know. All right, so photosynthesis, guys. Yeah, he's the one I taught him for the chemistry Olympiad actually, and then he made it, and then he went to I'm not because of that. He was a pretty smart kid actually. All right, photosynthesis, guys. So I don't know. I never taught from your school AP Bio, so I don't know, but I know how AP Chem is. So first, this guys. So this one, you know, so the time given, guys. So for you guys for the AP, so this one, guys. They don't go too crazy in the detailed perspective. So AP, right? They changed the whole theme, okay? Back in 2013 or 2012, I forgot when, okay? So even almost 10 years ago now. So what they changed to is they don't want you to memorize stuff, okay? Like the mechanism steps, okay? They want you to know the main idea, okay, and the higher level understanding of it. Like higher level means, guys, like not in detail, okay. So, but we'll see, guys. I'm not gonna get too crazy in detail for you guys, okay, with the photosynthesis. But I don't know. Depends on your teacher. Definitely not Jericho. Definitely not. But I'm a little worried about this guy. So let's talk about it, guys. Photosynthesis. So photosynthesis, as you can see from guys, synthesis. So when they talk about synthesis in bio, okay, what is synthesis, guys, by the way? Hey, what is synthesis, guys? Synthesis is what, guys?
Making.
Huh?
Putting two things into one.
So there's a better term for that in bio. What is that?
Reactants.
I would just no. So smaller parts into a bigger piece. What do we call that process in bio? Yeah. Anabolic. Very good, very good. It's called what? Anabolic process. Okay. The opposite is what, guys? Catabolic process. Okay, that's bio. So like in chemistry, we call it what, guys? Decomposition in chemistry, guys. You know, that's like synthesis. You know, like, but in bio, they call this what, guys? Anabolic process. Okay, and typically, right? Especially in bio. Okay, bio is like you know, it doesn't sometimes make sense in chem perspective, right? Typically, when we talk about anabolic reaction, okay, what do they need, guys? They need to put in energy for this process. A lot of times, guys, anabolic process is what, guys? Endergonic. Okay, endergonic. Okay, so which means what, guys? Okay, synthesis, which is anabolic process to produce glucose or G3P, whatever you want to call this. Okay, PGAL, whatever, however you want to call this, is what requires energy. This energy is what, guys? Photo, which is what? Light. Okay, so it's an anabolic process, okay, which requires the energy, which is an endergonic process. The energy is produced by provided by what? Overall, the light. Okay, light anabolic process. High level, very high level. Okay, so this is what, guys? G3P, glyceraldehyde 3-phosphate. Okay, that's a product of the cell's photosynthesis. Okay, this can be turned into glucose. Okay, so yeah, and that's how the plants will do it. Okay, plants will produce what? Glucose from this guy. So you know. But now, let's just assume it's glucose that's the final product here, okay? Let's understand this, okay? Let's see what is needed. So some of the folks, we talked about it, guys, okay? So let's talk about what's really needed for this process. So, photosynthesis, right? So let's talk about, guys, so the highlight of all, guys, for this process is basically what? Something we call Calvin cycle. Calvin cycle is what? Part of the photosynthesis light independent reaction. Okay, they don't call this a dark reaction. When I was in high school, they used to call this dark reaction. Okay, but they changed it to what? Light independent reaction. I'm not gonna write it. Okay, light independent reaction. That's called the Calvin cycle now. So keep in mind, guys. What do they do, guys? They bring in CO2. Okay, they call it, they call this what? Fixing of the carbon dioxide gas. And what happens, guys? They produce G3P. Okay, that's a Calvin cycle. So now, like, okay, so CO2 is what? The smaller subunits, okay, where they're turning it into what, guys? Larger G3P, it's a 3-carbon molecule, okay? So this is an anabolism process. That right there is what they call the anabolism process. Okay, this is the anabolism process. Okay? So as we discussed, okay, as an anabolic process, what do they need? What do they need, guys? They need what? ATP. Energy is needed for this process. Okay? Just like how we talked about. So Calvin cycle, right? There are 3 steps of the Calvin cycle. 3 steps. Okay? Steps of the Calvin cycle. First step is what? Carbon fixation. Very, very important, guys, okay, for you guys on the AP. Carbon fixation, okay, which means what? You gotta understand, guys, okay? CO2 is not a gas molecule that's really like, abundant, okay? Carbon dioxide gas is what? Like, people say, guys, so people say, what, carbon dioxide gas is what? Like, dude, it's a global warming and stuff like that, right? Yeah, what do you call it? CO2 is what we call as what? What do you call it? The greenhouse gas, right? Greenhouse gas, right? But people think, oh yeah, like we have a lot of CO2 in the atmosphere. Less than 1% is CO2. Less than 1%. Okay, less than 1%. If you go out, you see 100 people, there could be 1 CO2. Okay, one guy that's like what? Like CO2, like not even 1%. It's getting rare. Okay, it's a very rare situation to get a CO2. Okay, to find a CO2. Okay, so which means what, guys? There's an enzyme. Okay, this is important, guys. There's an enzyme called Rubisco. You gotta know this, okay? Enzyme called Rubisco. Okay, it's an abbreviation for something. I don't know what abbreviation it was. That fixes the CO2. Okay, brings in a CO2 because what? It's not a lot out there. Okay, think about it, guys. 70% is like N2 gases. Like 20% is like O2 gases, right? So like, okay, so there's like some argon gas, some other crap, and then what? And then CO2. There's not a lot. Okay, it's hard to find them, right? So which means what? Yeah, they got an enzyme for this. They got one job. This guy have one job. Okay, bringing CO2. So this is what they call what? The first step is what they call carbon fixation. Fixing CO2, okay? Bringing CO2, okay? There's one other thing you have to know, guys, like there's other like fixation-wise, guys, there's like nitrogen fixation you have to know down the road for your college chapter. But carbon fixation, more of a CO2. But even that, this guy's having a problem with this, by the way. So I explained this to you guys before, right? Like Rubisco. So keep in mind, guys, Rubisco is the most abundant protein that's out there. Okay, that's the most abundant protein that's out there. Okay, so like out of all the protein that's out there in the earth, this is the most abundant protein. Okay, they got the most one out of this. So all the plants will have it. All the plant cells will have it. Okay, by the way. But the problem is what, guys? When temperature gets a little hotter, what happens, guys? They don't bring in CO2. That's a problem. Okay, so then we'll talk about it, guys, probably next week. So, and then what? Keep in mind, guys. Okay, CO2 to glucose. like C6H12O6. Okay. So I'm not, again, you cannot, like I cannot teach you guys like chem here, but a lot of you guys came from AP Chem right now, okay? So if you calculate, okay, the oxidation state of the carbon on the CO2 versus what? The carbon over there, okay? Again, a lot of you guys come from AP Chem, right? Okay? Carbon, so oxygen's charge is what? -2, right? Like, dude, like -2 is the state of oxidation, guys. Carbon is what, guys? +4. Right? So, you know, charge is +4. What about this one? Hydrogen is +1, so there's +12 from these guys, and what? -12 from the oxygen. So carbon charge is 0. Do you understand, guys? So carbon is going from what, guys? +4 to a 0. What's happening to the carbon, guys? They are being reduced. Carbon is being reduced. Okay? Now, what does it mean by reduced, guys? They're gaining electrons. You understand, guys? They're gaining electrons. So you gotta understand, guys. Okay, so if you understand, guys, ATP is needed for anabolic processing body. Okay, so which means what? Where are they going to get the electron from? Okay, so next phase, there's the first phase competition. The next phase is what? Next phase is what they call reduction, carbon reduction. Reduction is the next phase of the Calvin cycle, which is what, guys? Getting this electron. Okay. So where are they gonna, so the big question of this process is what, guys? Who's gonna give them ATP, and where are they going to get the electron from? That's a big portion of this, okay? Calvin cycle. And then also, guys, I mean, so the third is what, guys? Third process is what, guys? So it's a cycle, which is what? They have to what? It's not really important, guys, right now for you guys. It's what? Regeneration. So fixation, reduction, regeneration, which is what? To run the cycle again. Okay, so you know. So the question becomes, guys, okay, where are they gonna get the ATP? Where are they gonna get the electron from? That's a question. So to run this process, okay, we gotta understand, guys. Okay, so where do they get it from? So are we gonna borrow ATP from mitochondria? No, we're not gonna. They're not gonna get it from mitochondria, just so you know. So, and the electrons, where they get it from, is a question you have to ask yourself. So so production of these two is coming from what we call the light-dependent reaction. So guys, this is chloroplast, by the way. Chloroplast, guys, looks like this. Okay, so chloroplast, I mean, you probably see, guys, like little sacs, you know, like this. So I'm not gonna show the whole thing, by the way. This is called the crana of the chloroplast. The fluid portion is called the stroma. Stroma and crana. Okay? And the crana is where, guys, this is where they have what, guys? This is where they have a thylakoid. So thylakoid, guys, is where they run, okay, the light-dependent reaction. Oh yeah, this one, light-independent Calvin cycle as well. Calvin cycle occurs at the stroma portion. Stroma is where this runs. Okay, and the other portion, which is like the thylakoid, right? This is what, guys? This is where they run what, guys? The light-dependent reaction. Okay, the thylakoid. That's chloroplast. They got their own DNA. They divide themselves. Blah blah blah. You know. Well, let's talk about, guys. So the light-dependent reaction, right? It occurs what, guys? At the thylakoid. Okay, that's what it happens. So what's on this thing? Just talk about it. High level here. Okay. So high level, guys. On the thylakoid, this is a thylakoid membrane. Okay. Listen carefully now. Thylakoid membrane has what? Something we call photosystem. There are two different photosystems, by the way. Photosystem 1 and 2. Okay. These photosystems have what, guys? Have a pigment called, you probably heard this, right? Chlorophylls. Chlorophyll is where you can find, guys, in the photosystem. Okay, chlorophylls, right? So these chlorophylls are found in the photosystem 2 and the photosystem 1. Okay. And now So they call this photosystem like 700, guys. They call this like photosystem sometimes like 680. And these represents the wavelength of 700 nanometers and 680 nanometers. That's where the wavelength, where most of the wavelength, or major of the wavelength is absorbed. 700 nanometers, 680. That's what? Around red light range, just so you know, okay? So how do they do this, guys? So if you look at chlorophyll, okay, chlorophyll, right? So chlorophyll has, chlorophyll is a molecule, by the way, okay? Which has a magnesium center. You have to know about magnesium center. Magnesium is in the center of a chlorophyll, okay? So it has a metal. Like, you gotta understand, guys, the shape of chlorophyll looks very similar to hemoglobin. What's on the hemoglobin, guys? Hemoglobin is what, guys? They got what? Iron in the middle, okay? Fe, right? But chlorophyll is a magnesium center, okay? So keep in mind, guys, having a metal inside the center of the molecule is what, guys? That means what? They have a lot of electrons that can be displaced. Electron can be popped out, okay? So basically, guys, it's surrounded by a lot of, like, magnesium, non-metals and stuff like that within the photosystem. So I'm not gonna get into like crazy detail right now, guys, for you guys. So basically, guys, at a high level right now, what will happen is, so we're understand, guys, so we need what? Electron. Okay, we need electron here. So, and also ATP. So what they gotta do is this. Sunlight. So sunlight, right? Sunlight, like if you look at sunlight, even this light, right? Is why they call this a white light. Okay? White light. Look at, like, the color is white, right? So we call this white light because what? It has every band of colors from what? Violet to what? This is like red. All the visible light, if you put that all together, it's what? It becomes this color. Okay? But now what happens, what, guys? So this guy, photosystem 2, they absorb around 700 nanometers, around red light. Okay, they absorb the best around here. Okay, so then what absorbs it is what, guys? The chlorophyll in the photosystem will absorb that 700 nanometer lights. Okay, so yeah, typically, right, from your chem, like, dude, from your chem, right? If electron gets excited by a light, what happens, guys? So there are a couple things that can happen, right? What do you think? Yeah, what happens?
It increases its physical energy level.
So yeah, like, yeah, it gets what? The light energy converts to potential energy of electrons, okay? So the energy of the electron can be turned to a couple things, right? Which is what? And the electron can be excited to the higher level, or what, guys? It can be ejected. You know what I'm saying? It can be what? It can pop out from magnesium. You know what I'm saying, guys, here? So basically what happens, what, guys? Okay, so the sunlight will hit the photosystem, it what? Ejects the electron, yeah? Electron ejection. So this is a very high level, by the way. It's like there's a reaction center, guys. I mean, that's a detail that I'm not gonna really go for the AP class, okay? So now, sunlight will hit it. So, okay, so sunlight will hit the electron, guys. Electron gets rejected, okay? So now, the problem is, okay? This electron is a high energy electron right now, okay? But let's talk about this, okay? There's a problem with this. Chlorophyll, they do not have unlimited electrons, okay? They have to get replenished by something, okay? So think about it, guys. Like you spend some money, okay? You don't have unlimited supply unless, like, I don't know, you hit a lottery or something. You gotta get back from something, okay? You gotta earn the money, right? So where are they gonna get it from? Question, right? Where are they gonna get it from, guys? Water, H2O. H2O. So H2O is there to resupply the electron lost by the chlorophyll, okay? So H2O, so you gotta understand, guys, they go through something called photolysis. So photolysis is what, guys? This is where, guys, water is turning into what, guys? O2 gas and H plus and electron. It's not a balanced equation right now. If you're coming from chem, guys, you should know this equation, guys, okay? This is what oxidation of water, okay? So now what happens, guys? The electron here replenishes the photosystem, okay? So, yeah, do you see this, guys? Yeah. Energy from sunlight, okay? Electron from water. You see that, guys? So now, so you can kind of see, guys. You probably heard this, right? Like, okay, what do you need for plants to grow? Which is what you need. What? Sunlight and water. That's all you need. Okay. Sunlight ultimately provide them the energy. Okay. We haven't really converted to the ATP yet. And what? So, the water provide them overall for the electrons. You understand what I'm saying, guys? That's all they need. Okay. So now is what, guys? Conversion. Okay. To the what? ATP now. So basically, guys, you know what I'm saying, guys? What's gonna happen now is this, okay? This for now. So what's gonna happen is this electron is what, guys? Now becomes what they call high energy electron. This electron is what? Have a relatively high energy now. So what happened, guys? So what they have here is they have these proteins called cytochromes, okay? So their job is to what? So using the potential energy of electron, okay, what are they doing is what? They are trying to produce, okay, electrochemical gradient of a proton. So high, like, you know, very fancy name for not much fancy stuff going on. So the electron will be accepted by the cytochrome proteins, okay? And what they do is, keep in mind, guys, here, this is a stroma. This is a thylakoid, okay? Space. So what they do is what? They use this energy of electron and what? They pump in a proton into the thylakoid space, okay? So the electron is being passed to one protein to the other and what? They pump in these proteins, okay? There's just what? By what? Active transport, okay? They're pushing it in there, okay? Using the energy of what? Not ATP, but what? The electron. Because the electron is excited by what? Sunlight. Okay? So overall, the sunlight energy converts to what? Converted to the potential energy electron. They're using that potential energy electron to what? For the electrochemical gradient of H+. Okay? That's what's happening right now. And then what happens, guys? The electron will be. So now, so they're pushing in the H+ into thylakoid space. Okay? And now, okay, so listen carefully, okay? The sunlight can hit this photosystem 680. Okay? So this photosystem, what, guys? It means that they can excite the electron here as well. The electron is excited. Okay? So you gotta understand, guys, what replenishes electron lost by this one as well? What do you think, guys? This one as well, guys. Yeah, the electron from photosystem 2. Okay, the electron from here can replenish photosystem 680. Okay, but now, so like, and what happens, guys? Yeah, so then what happens is they keep on pumping in these plus into the thylakoid spacing. Okay. So keep in mind, guys, what you're doing here is what, guys? Converting sunlight energy into what? ATP. That's what we're doing right now. Okay. And then what? What do you think, guys? So it's typically, guys, I mean, again, if you guys have heard of this process before, guys, how do they convert these H+ concentration gradient, like electrochemical gradients of the H+ into ATP? There's something they call what, guys? There's something they call ATP synthase. Okay, it's called ATP synthase. Okay, it's an enzyme that what can mass produce ATP, right? So, like high level, right? You understand, guys? So basically, what's happening is there are a lot of these H+ in the what? Inside the thylakoid compared to the outside. You understand what I'm saying, right here? All they do is what, guys? Using the sunlight and what? They push in these H+ into a thylakoid. Okay? And what happens, guys? This builds what? The electrochemical gradient. This is very important, guys, okay? It's called what, guys? Electrochemical gradient of H+. Okay, so now, you understand, guys, why do they call it electro and chemical? Electron is what, guys? Because what? It's a positive charged particles here, okay? It produces what? Electric potential, and also what, guys? Chemical means what, guys? The particles of hydrogens, okay? So they have what? It's not a chemical gradient, it's not an electro gradient, it's what? Electrochemical gradient of H+. That's a term they love on the AP, okay? So there's a lot of these guys in there. Yeah, what do you think they will do? You know what I'm saying, guys? First thing first, guys, keep in mind, the sunlight excite the electron. The energy of sunlight is what? Converted into potential energy of electron. Okay, excite the electron. And what happens right now? At that moment is what? It turns into electrochemical gradient of high H+. That's what the energy is converted into. Okay? And then what? This, how are they going to produce ATP from this? The job is ATP synthetase. That's the job. Right there. So what will happen is what? This very high gradient of electron H+, what happens, guys? There is a special protein called ATP synthase. Okay? Again, yeah, the function of the ATP synthase, the guy who figured this out won Nobel Prize for this. Okay? You do not have to know in detail how they function for AP. Okay? You don't have to know, like how they function. There's an F0, F1 subunits and stuff like that, alpha and beta subunits, F1, like nothing like that. Okay? You don't have to know anything like that. Okay? You just have to know there's something called ATP synthase. And what happens, guys? What they do is what, guys? They let these guys go out back into a stroma. Okay? By what? This is what, guys? This is what they call facilitated diffusion. Could you? Like that's what we talked about, right? I don't know if you talked about, but like facilitated diffusion, right? But they put a different name for this. What do they call this, guys? Chemiosmosis. They call this what? Chemiosmosis. Okay? So they're like, dude, they're gushing out, okay? And there's an ATP synthase. There's a function behind these guys to do what? Which can produce ADP, which can produce ATP from ADP and phosphate, turn this into ATP. Okay? This process is called what? Oxidative phosphorylation. Okay, this process produces ATP. So H plus goes out, it mass produces ATP. Okay, and this process is called oxidative phosphorylation. You gotta know this, okay? This is a process called oxidative phosphorylation. Okay, that's how they what? Produce ATP. Okay, by what? PS2 to PS1. Okay, you gotta understand it. It's very simple, dude. Like it's, dude. I don't like bio because I mean again, chem is the same thing. Okay, this is what it is. Okay, you gotta believe it. Like it's like reading a Bible, right? Like Bible says this. I mean that's why I'm not very religious, guys. By the way, they say this is right. That's correct. That's how we know. That's basically what it is. Okay, you gotta believe it, right? I mean again, dude, I'm not a very religious person because, like, how do I know? We're very scientific here. Like you guys are learning this. Same thing here, okay? They tell you this is how it occurs, okay? You gotta learn that's the process and what? Just believe it, okay? Same thing, okay? That's why I like the physics the best actually. Physics is like you throw a ball, you know when it's gonna fall, like it makes more sense, but it is what it is right now, okay? So this is how the process occurs and it's a high level though, right? But based on the oxidative phosphorylation, that's what happens, guys. So more to come on this. Okay, so we have to understand there's more to come on this because we have to understand the cyclic phosphorylation and non-cyclic phosphorylation, guys, and also production of how are they going to carry the electron for the reduction process. So how do they carry the electron down here? That's also what we have to discuss as well. But next week, guys, we're going to finish off respiration and photosynthesis next week, and then after that, probably next week we're going to do that, and the week after we're going to do the cell communication. That's the whole week. That's the whole class. And the week after, it'll be a cell cycle. Okay, and then after that, it'll be genetics. I think that's going to be it for us for the summer. Okay, I mean, if the time is remaining for us, genetics is a big topic, by the way. So I'm not sure, but then I'll touch on the DNA replications and protein synthesis. Okay. All right, guys, do your homework, guys, and I'll sign them, guys. Probably I don't know by Monday. Okay. All right, guys.